Breaking the wall of ovarian cancer diagnosis. Agnes Reiner, Universität Wien, Austria.
Over the last decades, the mortality rate of many cancer types has decreased, but that's not the case for ovarian cancer. And that's because 75% of ovarian cancers are diagnosed at late stages, leading to the fact that only 10 to 30% of the patients survive the first 5 years after their diagnosis. So an earlier diagnosis would be the solution, but how can we break this wall? By finding a crack in the wall. Extracellular vesicles are small lipid bubbles that are produced by any kind of cell. And what makes them so promising for diagnostics is that they look like the cell they are coming from, and also they circulate throughout your whole body via your blood system. So simply put, you could collect the patient's blood sample and then analyze whether cancer vesicles are present or not. That's why I've studied extracellular vesicles of ovarian cancer patients and found that they carry a very specific protein and lipid composition that can be used as a biomarker. This crack alone might not be enough to tear down this big wall. You also need the right instrument. So additionally, I've developed a hammer to tear down this wall. Plasmonic biosensors are able to detect their target analyte with extremely high sensitivity, and that's exactly why I've chosen this method for the detection of the vesicles. To also achieve high specificity, I'm using a two-step procedure in which I first bind the extracellular vesicles to magnetic nanoparticles based on the lipid composition. And then, in the plasmonic biosensor, I probe for their protein composition. Additionally to this, I make use of the magnetic properties of those nanoparticles, and by applying a magnetic field, I can highly efficiently collect the target analyte, the vesicles, to the sensor surface to increase sensitivity. So by these means, I was already able to detect extracellular vesicles from a model system with high specificity and high sensitivity. And in future, by combining extracellular vesicles and plasmonic biosensors, we can achieve to break the wall of ovarian cancer diagnosis.
Thank you very much.